I do have a question for Valeria, and the question is about the mineral material that you described in the guts of the chironomids. And I was wondering if that is something that goes and transits all the time like food, or if they have to get something and then it stays into the gut like inert material. This is the question I wanted to put.
Andrea, it is a very interesting question. Just to answer to you, I found some larvae with empty gut. So I think that the ingestion is occasional, and so this mineral material is not stable in the gut, because I found some larvae just without any mineral particles inside. So,
because the possible function I thought could be to increase the available surface for microbes to colonize the gut, because it's like having what you normally find in bioreactors in activated sludge, blankets, or even diatoms, just to involve Carlos also in the story. When you have
yes.
Industrial bioreactors to perform useful reactions, normally they can be filled with diatoms, not because of the diatom's life, but just for the
Yes.
surface offered by the inert mineral material. Or in other cases, you have even plastics and things. So something which has a lot of extra surface might serve the needs of improving the high efficiency of maybe microbial-driven reactions. That's a
Yes,
possible.
it is possible, yes. It's just a way to, an adaptive mechanism to consume more bacterial biomass, for example. So
Yeah.
just to offer an extra substrate for bacteria. It's possible, yes.
Yeah.
I'm waiting for the results
Yes, actually, I take the
of
chance
our
to.
analysis, just probably to understand even this thing. I don't know exactly why they are full of these minerals, but of course this might be a reason. Otherwise, if I think these minerals could be dangerous for the animals, they evolved in some way something to filter these particles, but they
Actually,
ingest a lot of these particles without any dangers for them. So
yes, and answering this and also your question on updates, one of the possible reasons for our slow pace in extracting DNA could be this mineral content. This is something that Luca Mazzon, colleague entomologist, is telling me, because he is now finishing the last 3 specimens, which were the live ones that we brought back and then
okay.
kept. And because he wanted to extract the guts nicely and neatly, but he's having a lot of trouble under the microscope, the stereo microscope, because the things are small and there are this inert material that normally you don't have in other in Coleoptera, for example, in other
Yes.
insects. And but now I gave him an ultimatum: just extract the DNA the way you can, and we can sequence when you're ready. So be patient and we'll get there. Then the other thing is to wait for sequencing runs and platforms, but we will do everything hopefully soon.
Thank you very much for
Yes.
this update.
Yes.
Can I ask a question to Sylvia? I think Sylvia, your research on perception of soil and humus with students at the age of 15 to 17 and so on, that's very interesting. but how do you see the problem of ambiguity of language, you know, and terminology, that this has been kind of influencing the outcome, you know, that people understand not normally what the scientist understands, you know, and so on. And I think that's a question how to teach you know, soils and science in high schools, you know, and secondary schools. I think that's a very important question. How do you see this aspect?
Silvia. Mm. Silvia, we can't hear you. It is right. It's right what you, Herbert. You put the finger in the. But we should be able, as scientists, to speak a language that is clear also to other people. And it is a pity that young person could not have the same behind the word of soil, that they have one meaning that is completely different from the ones of the scientists. We should find a mean to explain people that soil is a living system, and
I think it's a question of communicating our science, you know, that's the thing, you know. To go into teacher training and so on, you know, to do also this not very scientific, you know, extension work to communicate with the public, you know, and so on, because that's very important, you know. And I think it's manifests through what Sylvia has shown, you know, that there is a great gap between the soil sciences and what the public communicates and perceives, you know. And I think this holds tomorrow. This holds also true when it comes to climate change, you know, and so on, and mitigating climate change for tomorrow, you know.
But even we scientists, there are differences in soil meaning. Andrea said that soil tried to find out when we had the first soil on Earth. Huh? When he was speaking, I was thinking, but for me, the soil, the first soil, is the sub of the experience of Miller-Urey experience. This sub, in water, they put in water some organic matter, and then they try to have the first sign of life on planet Earth. And then in this ball, there was the first soil of planet Earth. And I don't think it is so. It's probably at the beginning of life on planet Earth. Not so far as you. So soil might be different even between us. We don't have the same meaning of soil.
Yes, actually, I think one topic on which we can agree or not is who makes soil. Because if you say soil grows plants, this is one vision. If you say plants grow soil, that's a completely reverse story, and is the one I consider the correct perspective. That's why I say there were no soils before plants exited waters and colonized lands.
I think that Carlos will say that plants also we have to decide what plant what plants is. So a first plant on planet Earth could be a microorganism too, without roots. No?
Well, it has to do photosynthesis, so it must be a cyanobacterium or something like that. But I would call plants maybe the higher plants. I don't know. I would call bacteria the cyanobacteria, the blue-green algae, and plants the rest, maybe from.
For example, Carlos showed, who's presented us, a sort of soil made of microsoils, very little ecosystem within the soil itself. I saw this on your picture, Carlos. You have very, very small ecosystems within a gram of soil. So this is soil or not? And this type of soil was present at the beginning of the life on planet Earth. Ah, Carlos, you are mute.
Oh, sorry. Oh, no. No, I was just telling about the cyanobacteria too. Well, the diatoms are relatively recent in the evolutionary history of these microorganisms. They are since the Jurassic, more or less. So they are already more evolved in this endosymbiosis theory. Yes. So we have different genomes for each of these components of this. Even though they are unicellular, they are quite complex already. I was trying to show the image that you wanted me to show, the second one, and how they are living in this small piece of soil here. I can.
It's not easy to find a good definition of soil, because it is connected to the process of death and this one. Okay.
We can, so this image is taken with this microscope that yeah, we are seeing directly the image. There is no coating, and it works in a low vacuum, so we can see the structure. This is really the structure of the cells. I think probably this not roots, but it's probably like fungi. So we can see this line. So the diatom seems to be attached. Sometimes some of them are attached to these structures, but I don't know how they relate. I mean, because the diatoms, they are alive and they are able to move as well. If there is enough humidity, they move around. So they also can be not static. They go search for light. So they need light, so they can survive at lower depths as well. But yeah, it was for me, it was the first time I saw like this. So I was also surprised to see the amount of cells that I could find. But yes, they are basically the part of the soil. I think when we measure the silica content in a soil sample, if you don't distinguish biogenic silica or just silica from the environment, a big part of it will be diatoms. If we don't do this separation. But I don't know, I'm not a geochemist. I'm just wondering if there is someone who knows how to, or have experience with this kind of analysis, how important it is to separate these two type of silica and to measure productivity as well. So
Roberto, for example, Roberto, have you an idea about when your tardigrade came on planet Earth? At which time?
Can you hear me?
Yes.
Okay, so I mean, tardigrades on the sea, they arrived during Ediacaran, so before Cambrian, so before 600 million years ago. so before arthropods, and the exact period is not clear because they calculated with the molecular clock, but is during late Ediacaran period, so more than 600 million years ago. And there is a study by Rota Stavelli as the first author that tried to understand which they colonized Earth, and I think that it was I don't remember right now, but there is some. So they tried also to understand when they colonized Earth. So to see from the step from the sea to the Earth, and probably from our knowledge, the step they exit from the sea, and they probably directly went to Earth without passing through fresh water. So there was a direct step from the sea to the Earth.
Why do you think that they were first in water?
Because I mean, there was no environment for them 600 million years ago. So there was no life on the Earth at that period. There's nowhere. All the life was on the sea.
They were bacteria were there from 2 billion years still, at least.
Yeah, but no animals. There's no any traces that any animals colonized Earth at that period. So it's very hard to think that they were able to colonize Earth on that period too, because also probably the environmental conditions were not so good for them. I don't think that they can eat bacteria. I mean, there are species that eat bacteria and fungi, but I mean, you have to think that the first traces of colonization by animals are after the Cambrian. So they from molecular clock they were present before the Cambrian. So we think that the sea is the place in which they originate.
Massimo, are you still there? Massimo Tildo? No? I have a question. Is it possible to distinguish old DNA from present day DNA? Even trying to find out, to make some full shotgun metagenomics or in very old rocks and so. Is it possible to see if DNA is old or young? Or Andrea?
Well, what I know is that obviously DNA can be degraded in time, so ancient DNA is hard to isolate in long stretches. But for bacteria, DNA is protected within cells. And so if you are analyzing old sediments, you can find bacterial cells and extract DNA that you can sequence, and the result will look like modern live bacteria, but they might be mummies, they might be relics, just DNA which is preserved within dead cells. So soil or sediments are in this way like a museum of bacteria who are not necessarily alive, along with some which are still alive. So it is complex to disentangle the real active colonizers or survivors, physiologically active members of a substrate, a sediment or anything, from the ghosts, from the DNA from the past when it comes to bacteria. I actually had another question for Roberto about the temperature records. It is amazing that the tardigrades can stand 151 Celsius, I think I saw in your slide. And technically, since water is boiling above 90 degrees, how can you keep water liquid when you are going over 150? It's a matter of pressure. How do you do those experiments?
No, they can survive at the temperature only when they are dry. So there's no water inside the animal.
Okay, so there's no, sure.
When they are dry, also the temperature, they can survive at that temperature when they are dry, or even frozen at low temperature.
Right.
But that high temperature only when they are in dry condition.
Okay, so they can get completely dry. If you are heating at that temperature, they get life.
Yes, they lose almost all the water. Also the water that is in the structural water, that the one that is needed to keep the three-dimensional shape of molecules, large molecules, the proteins or TNAs.
I have the feeling that they look like animals, like little bears or things like that, but they are not so animal. They are more primitive things, more like bacteria, but they can take a shape that makes us illusion of animals.
They are a colony of bacteria.
Right,
They are not,
which is amazingly resistant.
yeah, but I mean, they are not the only one. I mean, there are rotifers and nematodes. There are species that can have that withstand similar conditions. I mean, there are, and also not all tardigrades are able to withstand these extremes. There are degrees of adaptations according to the environment in which they develop. For example, in freshwater environments, they usually are not so able to dry out completely because they don't need it. Usually they can stay in a very high humid environment. So instead, those that live in mosses or lichens, they are very good anilobians.
There is also one chironomid, is Polypedilum vanderplanki. It's living in desertic ponds in Africa, in the Sahara Desert. And it has very similar strategies to survive desiccation like tardigrades. And so from an evolutionary point of view, it's in a higher position respected to tardigrades, but the strategies to face the desiccation are the same, more or less. So it's common in several invertebrates, independently on the evolution of the taxa, so the positioning in the life, the tree of the life. I have a question for Roberto regarding the experiments you did with increasing temperature for tardigrades. Which was the range? Because the maximum, if I understood well, was only 5 degrees.
Yes, it was 5 degrees, 0.5. The maximum temperature increase within the chamber.
But this was the increase in respect to which temperature? The natural environmental temperature or the optimal temperature of each taxon?
The external temperature. So there was a system that record the external temperature and they increased the external temperature of 5 degrees, 5.5 degrees, all the 5 years round.
But regarding the daily variation, natural variations of temperature or seasonal variations of temperature, it was possible to reproduce, to mimic this. So inside your, in some way, mesocombs, you mimic the environmental variations in terms of the daily variations, seasonal variations, at any time 5 degrees higher in the mesocosm?
It was within the forest. I mean, it was an open chamber within the forest. So they created this chamber around trees within the forest. So the temperature that was inside this chamber was increased of 5 degrees respect to the temperature that was outside.
Okay, but anyway, changing during the day, even in the chamber.
Yes.
Okay.
So there
Not
was a
constant.
record.
Okay,
No,
okay.
no. That means the temperature drop of 10 degrees. For example, the drop at 10 degrees, within the chamber was 10 degrees plus 5.5. If the temperature increased at 20 degrees, in the chamber was 25.5 degrees. So it
So the
was
same
5.5
variation.
degrees old. See,
Okay,
change
the same
according to
variation.
the outside temperature.
Okay.
And I
Thank you.
just want to point out that your chironomids, the polypedilum, is the largest animals that is able to dry out completely. So
Yes.
this is there's no other animals that are so large that
Yes.
can dry out completely and survive. So it's something I think quite important to stress.
Yes. Yes. I have a curiosity for Carlos regarding diatoms on the glaciers, because we are not able to find diatoms in our samples collected in the cleoconites, for example. We recorded only cyanobacteria and some green algae, but no diatoms. So I don't know if you have experience on glacial diatoms,
No.
not just
No,
streams or
never sampled myself. No, but in this case, you were not sampling the rocks, right? Really, only the snow.
no, just the snow, the ice, but the bryce, mainly alloctonous, present on the surface of the glacier, and in the water running in the so-called barriers, so the channels forming on the surface of the glacier. And they are alive because we have tardigrades, chironomids, just on the debris, collembols, and so on and so on. Regarding the algae, we have the Camesiphon polonicus, for example, others, but no diatoms.
No diatoms. It's surprising. I don't know. I would say that they should be there. I don't know. Perhaps
but we have
sometimes
to check more samples, so it's not the final conclusion, of course. So just to know if you have information about that.
No, but they should be there. I mean, yeah, no, they are there. There are studies in high altitude. Of course, there are the richness of species is lower. This is proved more or less with the altitude gradient. It applies for diatoms. Yeah,
because we didn't find them even in the gut, and generally, if diatoms are present, most of chironomids eat diatoms.
Yeah, yeah. I saw one of your picture. I think that the third picture that you showed just when you were telling this, I mean, it was full of diatoms, really colonies.
Yes. Yes, but in that case, it was not. It was a special habitat, just in transition between hypocreal and glacial because fed by a vanishing glacier. So in that case, we have stones just covered by thick biofilm with a lot amount even of diatoms. So completely different habitat in some way. So transparent water, stable substrate, and so favorable, of course, for all algae in general.
I know that
gut content of glacial chironomids, no diatoms.
No diatoms. I know that there are in Antarctica they study the glacier diatoms. So there you can see thick lines of algae colonizing the glaciers, but in the Antarctica, in the Alps, I'm not sure if I have seen any article. I cannot recall now.
I heard also that diatoms can be transported, so like a drift, so that diatoms present on the glacier are not just from the glacier, but transported from another habitat. So this is another idea. I don't know.
Yeah, even the atmospheric deposition. You have seen this Sahara sand that was brought to Europe recently. It was completely full of soil diatoms. Full of, yes. So
So we have African diatoms.
Yes.
In the
And
okay.
And they are able to fly away, but leaving cells that colonize again and then leave again. It's they are not dead.
Survive? But can they survive?
Yes.
Just the
Yes.
African.
Yes.
Okay.
Mm-hmm.
Oh, wow.
Yes.
So Andrea and Augusto, we have to look for African diatoms in the gut content of this larvae. Very nice.
Augusto, I think Silvia wrote on the chat and also that her microphone might be working.
Yes, I think it's fine now. I don't know if is this the answer, but in Italy there is a lot of they teach at school a lot of specific words. So I have seen that from primary school they teach soil profile, soil horizon A, horizon B, C. It's really, but and also go on. They learn a lot of also in the high school, but the problem is that they read just and they don't deeper the knowledge, so it just a name, just a name. And the problem is also that the teacher that teach soil perhaps is a biologist or someone that in the doesn't really have contact with soil and don't know what is it or has a contact of soil in some sector. The same for a geologist that has to teach the human body. In Italy, we have the possibility of the scientists that can teach science for in 360 grade. So, but for me, the problem is when I was submitted this questionnaire that they feel that soil is just what I work on. Is this and more the answer was more like this for a student of scientific high school. And this is very bad because from hotel school they were thinking about what is and gives also, okay, they have ecosystemic value, and but the other one just okay, soil. I don't know if you have another other information regarding this, or for all of you, if you think the question must be, if there are some questions that are better than other. I don't know if you have in mind something.
Yeah, I think
Silvia,
it's hard to say because first of all, I think there is in teacher education, as you said already, there is some let's say some misconception and some let's say blind spots, and secondly, but this is also very important to communicate it, to have a clear and targeted wording, which is communicated also with the public, not because I think it comes from sloppy a gesture or a sloppy handling of the whole system. You know, soil is something we stand on. For sure, it's something we stand on, but we can also stand on concrete and so on and such things. You know, that's the thing, and we should kind of concentrate to get also a clear scientific message when we speak about soils, you know, as a scientific subject. This is not concrete and this is not something we stand on, but it is, you know, some living system, you know, where minerals, this and this and this interact, you know, an ecosystem. Yes.
And Silvia, I
Okay.
can add
Thank you.
something about the misconception of what soil is, because it's not restricted to young kids at school, but also people doing agriculture have a completely different concept if they are into conventional agriculture or regenerative, conservative, organic agriculture, because the former would say that soil is a commodity. Land is something that you can grab, and soil is a substrate in which you can put your seeds, and it is supposed to give you plants. The opposite is true. The plants are maintaining soil. Plants are releasing organic soluble compounds that microbes are processing and turning into complex structures of organic matter, which are at the basis of soil stability against erosion. So when you are plowing soil or doing mechanical chiseling and all the operations that are common into conventional agriculture, you are just bombarding an underground city made of fine cathedrals of organic matter that people are completely neglecting, ignoring soil microbiology and the role of microbes in soil making and processing of plant exudates to stabilize soil. So it is not a matter of lack of definitions, but it is a completely different view of what the world is and what the future will be if you are not understanding what soil is.
What I think is that for us that we are soil scientists, that we must give the a knowledge more a bigger knowledge on the soil, because otherwise it's difficult to save it, or to say is important. Why is important? It's just soil, but so it's, you know, I think we have to do more in this field, also with other people, because I was working for an article with a geologist, and he was thinking about soil that was something that was cover the rocks and was very bad for so the approach is completely different also. And but I think all the works that I have seen just today, Yatume, and all this is all together. We are very different, but all of us says that soil is important. So
but you see,
we must find something.
I
Yes.
think that some of the, let's say, mistreatment and today's problems we have with soils, you know, is coming from this sloppy and not correct interpretation. You know, especially that we have a certain carelessness when it comes to sealing soils converting fields to built over areas and so on, converting them to roads and so on. You know, all these things come from this you know, kind of ambiguous communication. And I think it's not only the students in high school and secondary school. Most important is journalism. You know, in journalism, the communication is very bad and so on. And so the people are careless and reckless when it comes to handling soils, you know. Good.
Thank you.
Thank you.
Augusto, you are mute. Your microphone.
I think that little by little, thank you, Andrea, people understand that soil is a biological ecosystem. And I see students and the fact, even the fact that we are going into a more biological agriculture, for instance, or organic food production, this means that people understand that soil is important. We can go on with the discussion tomorrow, I think, unless people will, because we had a great afternoon, and tomorrow we will continue in the same spirit, in the same. And I thank you very much for this. I was very happy with you. Ciao. Let's see you tomorrow.
Ciao.
Ciao. See you tomorrow, guys. Ciao,
Ciao a
Daniela.
tutti.
See
Bye
you
bye.
tomorrow. Bye bye. Ciao, ciao.
Thank you.
See ya.
Bye bye.
Ciao.
Ciao.
Bye.